The NIH Center for Computer Integrated Systems for Microscopy and Manipulation, or CISMM, serves over 20 collaborator and service projects across the country. We're located in the University of North Carolina in Chapel Hill and occupy over 5,000 square feet of laboratory space in the adjoining computer science and physics buildings. The CISMM Software Development Lab, located on the second floor of Sitterson Hall, houses computers running the operating systems our software is released for, including Apple's OS X, Linux, and Windows. Some of these systems have stereo displays, which produce greatly improved perception of 3D shape. Graduate research assistants in visualization and image analysis work in this space, developing open source tools for collaborators studying cancer, clotting, and cell mechanics. The lab also includes force feedback displays for the development of our custom atomic force microscopy tools, such as the nanomanipulator, which became a commercial product, and our new Prism side view microscope system. Our stereo and force feedback displays, combined with a foot pedal, enable physicians to plan surgeries for infants using our virtual pediatric airways workbench. Collaborators often visit our lab to use these specialized capabilities. We also provide online instructions for installing them in their own labs, both at UNC and other institutions. Finally, the lab includes a prototype control system for our 12-way parallel microscope. This enables debugging on the Texas Instruments-based microcontroller system we use to provide microsecond-level control over cameras and lighting. This space also serves as a location of our annual software workshop, where participants from across the country come to learn how to use our software and hardware systems. Its 10 development, display, and analysis workstations, together with our file server and backup server, enable CISMM personnel to develop tools for our collaborators. Our web server enables us to disseminate them to biomedical researchers across the U.S. The oscilloscopes and other electronic test and measurement equipment required for development comes from the Applied Engineering Lab, located just down the hallway. The AEL provides engineering support for computer science projects and has been heavily involved in all of CISMM's hardware development efforts. It continues to be a resource for our 3D magnetic force instruments and our Panoptes 12-channel microscopy system. The lab has an inventory of analog and digital signal generation and signal analysis equipment, as well as soldering stations and components needed for design and repair of the custom control and measurement systems developed within CISMM. Many of our undergraduate and graduate students receive training in hardware and electronics in this facility. Together, the CISMM Design Lab and the Applied Engineering Lab continue to provide the capabilities needed to develop custom microscopy systems, such as our nanomanipulator, our 3D magnetic force microscope, and our Panoptes 12-way parallel microscope systems. The CISMM software developed as part of our collaborations has been disseminated widely via more than 150,000 downloads, but our collaborators' largest simulations run on the Biomedical Analysis and Simulation Supercomputer that is located in Sitterson Hall. This $2 million system was provided by an NIH High-End Instrumentation Award to CISMM PI Taylor. It provides 452 computing cores with a total of 1 terabyte of RAM, coupled to 180 CUDA-capable graphics processor units. It runs large-scale simulations for our collaborators in cell mechanics, pediatric airways, and clotting. 3 of the major user groups for the system include CISMM and our collaborators. The CISMM Science Labs are located right next door in Phillips and Chapman Halls. The CISMM Biology Prep Lab in Phillips is where we prepare cell specimens for our biophysics experiments and soft substrates for traction force experiments. Cardiac myocytes, bladder, ovarian, and pancreatic cancer cells are maintained in the cell culture incubator, then plated in the biologic safety hood. Essentially, all of our students and collaborators are trained in this lab in bead and cell methods. Cells and other reagents are stored in our standard or -80 freezer. 4 microscope stations are used to monitor cultures and to validate transfection and staining regimens. The computer workstation is used for 3D stereo viewing of fluorescent cells using our ImageSurfer 2 software and for data analysis including our video spot tracker. The CISMM experimental labs are located in the same building as the Chapel Hill Analytical and Nanofabrication Laboratory, or channel, a $7 million, 5,000 square foot user facility that provides a wide range of state-of-the-art analytical, fabrication, and imaging instrumentation. 4 full-time staff members maintain the equipment, train, and consult for researchers. Among other uses, CISMM researchers fabricate microfluidic systems using semiconductor processing equipment and a focused ion beam microscope, and collect images on channels transmission electron and environmental scanning electron microscopes. This laboratory is used for materials, substrate, and device processing. Here is a workstation for the preparation of the actuated surface attached post, or ASAP specimens, that are used in the clot diagnostics project that is joint between CISMM and our startup company Rheomics. Moving past the usual array of hoods and an incubator for lung cell cultures, we come to a microfluidics fabrication assembly station for our clearance-on-a-chip microfluidics system, which we use to study mucus clearance within the virtual lung project. We have built a rotating microscope platform so that we can image, at high resolution, the ability of beating cilia within the human epithelial cell cultures to move mucus upward against gravity, providing critical insight into the role of propulsion and biochemical adhesion in successful mucus transport. This adjoining laboratory is used for the electrochemical deposition of custom nanoparticles and nanotubes for drug delivery studies and for the ASAP technology. In this corner of the lab is our farm of genetic variants of Chlamydomonas, an algae of unicellular flagellates that we use to study the basic principles of cilia actuation within the virtual lung project. Our combined atomic force microscope and fluorescence microscope system is one of 2 systems located in sound-isolated rooms in our basement laboratories, providing excellent vibration isolation. The Asylum MFP 3D Bio sits atop an Olympus IX71 inverted scope with a Hamamatsu Orca flash camera capable of 20,000 frames per second in region of interest mode. The entire system sits on a Hertzand vibration isolation system and is enclosed in an acoustic hood providing noise levels under 10 piconewtons. This system is central to our collaborator-led projects investigating the mechanical properties of cells, cell nuclei, and blood clots. Our PRISM sideways microscopy system will be integrated here to provide both sideways and in-plane fluorescence imaging with simultaneous AFM-based mechanical measurements. The PRISM is brought into the region of interest by an independent micropositioner. This system is being developed primarily for cell mechanics investigations. For example, we will use this system to measure strains transmitted from the cell exterior to the nucleus and chromatin to understand how force-induced chromatin alterations affect gene expression, differentiation, and metastatic properties in cancer cells. The neighboring lab is also isolated from sound and vibration, with its control computers located behind the right-hand wall. It contains a Nikon TE2000 inverted microscope on a vibration isolation table with a high frame rate Pulnix camera and a high sensitivity photometrics Cascade EMCCD camera for low light fluorescence. The laser sources and optics behind the microscope enable both laser tracking and laser trap experiments. This system is a workhorse for 3D force microscope experiments in cancer cell mechanics and for rheology experiments in cystic fibrosis, biofilms, and clotting. Adjacent to these labs is Chapman B50, which houses a variety of analytical and imaging instrumentation. The CISMM Hitachi S47 SEM is used daily by CISMM researchers and collaborators to image magnetic beads, nanoparticles, and our actuated surface attached post technology. For understanding our force measurements, we study the anchoring and geometry of the beads on cells and cell cultures. The ASAP project makes use of these resources for development work. A startup company, Rheomics Incorporated, leases space under a facilities use agreement to co-develop and commercialize our clot diagnostic technologies. This room houses 2 3DFM magnetic tweezer microscope systems and our high-throughput systems, Panoptes and Monoptes. One 3DFM system with a nanometric stage on our spinning disc confocal microscope is used for cancer and endothelial cell mechanics experiments. It sits next to Panoptes, our array microscope system. Next to Panoptes is its smaller brother, Monoptes, which is a single-channel optical microscope that employs identical subsystems. The second 3DFM system, with full environmental control, is used for endothelial cell mechanics, drug delivery, and myocyte projects. We use our high-throughput array microscope system, Panoptes, to study mucus rheology, clot permeability, and cell mechanics. The system can perform experiments with complete data analysis on 96 specimens in approximately 6 hours with no user attention. The entire system, designed by CISMM engineer Leander Vici, includes 21 custom electronic boards, 15 of which are housed in an electronics box that controls signal synchronization. The remaining custom boards are located within the Panoptes base and provide camera control, sample illumination, Z-alignment, and positioning. 12 40x objectives located underneath 12 wells of the multi-well plate add fine, vibration-free control of the imaging plane through the use of liquid lenses. A nanometric stage moves the multi-well plate in X and Y, visiting all of its 96 wells in 8 steps. 3 stepper motors support the nanometric stage and provide fine approach and tilt adjustment that makes the plane of the multi-well plate parallel to the plane of the objectives. The experiment begins with a well layout program. This lets the user input metadata for each well and specify the parameters for executing the experiment, including the camera and illumination specifications, the number of fields of view to visit per well, and the well-to-well trajectory through the plate. The experiment then proceeds with Panoptes image acquisition. Typical parameters use 2-color epifluorescence video capture at 55 frames per second with 1 minute of video in each of 5 fields of view per well. This automated data acquisition step typically requires less than an hour. When finished with data collection, the system analyzes the acquired video. CISMM's Video Spot Tracker software identifies and tracks particles, producing a time series of positions for the beads in the fields of view. Tracking the nearly 1 terabyte of video requires approximately 5 hours for a typical experiment, producing trajectories for about 5,000 beads. Finally, the particle trajectories are processed by statistical analysis programmed in MATLAB. All steps, from acquisition through analysis, take about 6 hours to complete. Panoptes automates this entire process through computer control, making it a fully autonomous, high-throughput microscopy system. The 13 computers that serve Panoptes join another 52 computers that provide the computer-integrated systems for CISMM's unique hardware and software, enabling discoveries for the diagnosis and treatment of cancer, clotting disorders, and of pathologies of the airways.